Okay, students. So, good morning, class. Today we will be talking about the plasma membrane, right? We will be talking about its basic structure and function in living systems. I mean, what is its basic composition? What are the different biomolecules which impart different properties to this membrane? And then we'll talk about the role of these different components, these different biomolecules that form the plasma membrane, how they perform or how they help actually membrane perform the different functions. You can ask any questions in between. If you have, just type your questions in chat and I'll reply to that. So let's begin. So I'll first introduce you with the membrane, right? So plasma membrane, first of all, it actually separates the living cell from its surroundings, right? I mean, when we have talked of the structure of cell, right? We said that cell is the basic structural and functional unit of life, right? And in if we talk of the multicellular organisms, first of all, you see they have different tissues and organs which are formed of multiple cells, right? They are a combination of multiple cells. And despite working together, forming a tissue or forming an organ, they still behave like one. And at the other end, they are still autonomous and independent. This is all because each and every cell in that system has its own boundary and it takes care of itself, right? So this membrane actually gives the cell its proper shape, right? Its proper boundary, and I mean, it allows the cell to be a part of its surrounding also, as well as it separates it. It gives that particular, you can say, structure its own, you can say, protection from any adverse condition which is present in the environment, and at the same time it also helps that particular cell to connect with its environment also. Now once we think of the membrane, right, I always say we can compare the membrane to the outermost boundary of, you can say for example, your house, right? So when we compare the membrane with this example, it is actually to make you understand that the membrane is actually very protective in function, right? But it is not as rigid as the you can say walls or boundaries we talk of or we compare it to. Once you learn about its structure and function, you'll see it is rigid, it is protective, but at the same time, it is very active in communication of the cell interior to its external environment or cell outside. And it is all because of the very beautiful and special structure of the membrane, the distribution of the different components that we'll learn to. So let's see. So plasma membrane actually separates a living cell from its surroundings. If we talk of its thickness, approximately it is 8 nanometer thick, and it also controls traffic of different molecules into and out of the cell. Now, when we say it is controlling the traffic of molecules from cell interior, that is from cytoplasm, to the extracellular side, that is the cell exterior, or from the extracellular side to the cell interior, right? It does not mean that, I mean, it is freely permeable to each and everything. Anything can enter in a cell or can exit. First of all, different cells have different levels of permeability for different type of molecules. Okay, because their properties are different, their requirements are different. So, plasma membrane allows the movement of molecules in and out of the cell, but it is highly selective. Okay, it is selectively permeable. Selectively means it is very selective in what molecules can enter or exit from the cell, and permeable means it is not like that, it is very rigid. So it is permeable but allows movement of very specific substances to cross it. Now, the major components that actually form this membrane are lipids, proteins, and some carbohydrates. These are the three basic molecules that are organized in different compositions in different living membranes, right? And that impart different properties to the different type of membranes. Now, if we talk of the lipids, then the membrane is actually made of phospholipids, right? It is formed of different type of phospholipids, and it is a bilayer of phospholipids. I'll just show you a diagram so that you can understand. Okay, I'll just zoom it. Okay, so these are actually the phospholipids. Okay, you must have learnt about these phospholipids in class 12 also, once you must have been taught about the fluid mosaic model, right? So these phospholipids are actually the lipids that carry a phosphate head group. Here you can see this is one layer of the lipid and this is the other layer of the lipid. And this circular gray part represents the phosphate head, and these tails are actually the hydrocarbon tails. So you can see this is one layer of phospholipid and this is the other layer of phospholipid. That is why it is called a phospholipid bilayer. So is this clear to all of you, everyone, that what is meant by a bilayer? Okay, all right, let's move ahead. So, it is a bilayer of phospholipids with polar heads, which are hydrophilic in nature. Hydrophilic means water loving, and hydrophobic means that is water repelling or water one that hates water, right? So, once you saw those phospholipid structure, there you have seen that the phosphate head, which is represented by a circle, it is actually hydrophilic in nature, whereas the tails are, since they are hydrocarbons, so definitely they are hydrophobic. So this means these phospholipids are amphipathic in nature. That is, they show both hydrophilic and hydrophobic behavior. Now again, I'll just move to this diagram. Now you see, this yellow part represents the interior of the cell, that is the cytoplasm, right? Whereas this part, this blue part, is actually representing the extracellular space, anything that is outside the cell. Now, if you take a look at this membrane, it is a bilayer of lipids where you see of both these layers, the phosphate heads are extended towards the both you can say boundaries of the membrane. So the phosphate heads from one side, that is the hydrophilic end from one side, is actually facing the cell exterior, whereas the other end is facing the cytoplasm. And if we talk of the interior of the membrane, so all these phospholipids are the hydrocarbon tails of the phospholipids, which are hydrophobic in nature. They are extended inside the, you can say, in between these two hydrophilic ends. So this makes the interior of this membrane highly hydrophobic. So that is why any hydrophilic molecule is not allowed to pass through it. Any large hydrophilic molecule cannot pass through it, right? But if we take a look at the behavior of these lipids, then it has both hydrophilic and the hydrophobic part. So is this clear to all of you? If you take a look at this diagram, do you get this? That the extremes are hydrophilic, whereas the interior is highly hydrophobic. Okay, fine. Now the membrane is represented as a fluid mosaic model. Okay, so can you tell me why it is said to be fluid and why is it is said to be mosaic? Please post your responses in chat. I'll just wait for it and then I'll explain you things. You can take a look at this diagram also. I'll just slightly zoom it for you. Okay, and you can let me know, why do you think it is called fluid and why is it mosaic? I just want to see how many of you remember this. Okay, Shraddha says due to its flexibility and permeability. So, Shraddha, do you want to say that the fluid part represents the flexible property of the membrane and the mosaic part actually represents the permeability? Is it so? Okay. Let me just take a look at the responses of other students also, and then I'll tell you. Okay, Aman also says it is fluidity and flexibility. Okay, I'll just modify. Do you know the meaning of the term mosaic? What is a mosaic? Okay, Palak says flexible as it makes a barrier. Okay, fine. All of you. Okay, now I want a response for mosaic also. Please post. Fluid, all of you, or I mean those who have posted response, you all got it right. It is said to be fluid because it is not rigid, as I have told you. It is flexible in nature, and its components, these lipids, and here you can see these are the proteins, these are the carbohydrates. You can see the carbohydrates are either attached to the lipids or they are attached to the proteins also, right? These lipids and proteins, these lipids can move within the membranes, within the layers, and I mean they can change places, and it allows movement of molecules across it. So that is why it is made up of fluid. And Shilpa, now you got it right. A mosaic means one which is made of different type of components. A mosaic means something which has different combinations of components. So if you just take a look at this diagram, you can see here that there are different types of molecules in the structure of the plasma membrane. You can see the lipids here, right here you can see the proteins. This is cholesterol, and in lipids also you'll see that there is a variety of lipids are there, right? And if you take a look at the carbohydrates, you can see these carbohydrates again are of different types. Their length, their branching, their chains, they are varying, and these proteins also are different in structure as well as in function. So that is why this membrane is said to be a mosaic of different components, plus it is fluid in nature, means it is flexible and allows movement of molecules across it. So a fluid environment with a mosaic of proteins and carbohydrates which are embedded or attached, that perform or serve several functions, right? So now if we take a just close look at this membrane, here you can see a bilayer of phospholipids. Then if you take a look at the proteins, right, you'll see there are some proteins which are able to cross both these bilayers, while there are some proteins that you can see they are embedded either in the internal or the external phospholipid bilayer. Okay, so these proteins are called transmembrane proteins or integral proteins that can cross the lipid bilayer, and these proteins actually connect the cell interior to the cell exterior through the plasma membrane, right? And then you can see the carbohydrates, they are present only on the extracellular side of the lipid bilayer. The carbohydrates are never present towards the cytosolic side of the lipid bilayer. This is because I have told you these carbohydrates perform different functions. First of all, you can see all these components that you can see here in the extracellular fluid, they form the extracellular matrix that is called the ECM. Okay, so these carbohydrates, they extend towards the ECM side or towards the ECM of these membranes, and they are like address tags. They give identity to that particular cell. So the pattern of carbohydrates, I mean the way the carbohydrates are attached to different proteins to form glycoproteins, or to the lipids to form glycolipids. This pattern is called glycosylation. So the pattern of glycosylation, first it protects these membranes from different proteases, different pathogens that can secrete enzymes that can degrade the membrane and disrupt the integrity of the cell. And on the other hand, they also give recognition to the cell. Okay, like you can differentiate between two similar houses. Suppose they are just adjacent to each other. If you take a look at the nameplates and they have different names of the owners represented on them, you can just read and understand if you go there for the first time that to which house you want to go or which you can locate. So it simply means that it represents the identity of that particular place. So in this case, this pattern of glycosylation actually gives the cell its identity. Okay, so is it clear to all of you? What are the different components here? Okay, any questions on this? Okay, Diksha, do you have a question? Okay, I think you do not have any questions, so let's move ahead. Now we take up all the components that we have seen in that diagram, in that picture of the lipid bilayer or the plasma membrane. We'll take up all the components one by one and see how their composition varies in a plasma membrane. So first of all, since it is called the lipid bilayer, the plasma membrane is also named so, we'll talk of this lipids of the membrane. So these lipids actually form 50% of the total membrane mass. Then they are aliphatic in nature, which I have already told you that they have a hydrophilic phosphate hedge and a hydrophobic hydrocarbon tail. Then these lipids are self-sealing. You know, lipids have tendency to self-seal. So they actually, appear to be, they can, I mean, they make the membrane very protective through this self-sealing property. Okay, I think Palak has a question. Okay, Palak did not get about the Mozart part we have talked of. Okay, so I'll just go back to it again. Okay, Palak, I'll take you back to the diagram. Mosaic, as Shraddha told us, is something that is formed of different components that are not same. You can see a variety of components in it. So if you take a look at this diagram, you can see the membrane is a mosaic of different type of components. Especially if you take a look at the proteins, right? Uh, Once I'll talk of the membrane proteins, you'll see they are structurally and functionally very different, right? Very diverse, very different. So that is why it is called a mosaic. So it is different kinds of proteins that are different in structure and function. So hence it is called mosaic. Okay, Palak, is it clear to you now? Palak, can you hear me? Okay, so we were talking of membrane lipids. So I told you these are amphipathic in nature, these are self-sealing, okay, and then these lipids actually contain, they are phospholipids, then they have cholesterol as well as the glycolipids. So these phospholipids you have seen in gray color, in that diagram. Then cholesterol was represented in yellow, and glycolipids, you must have seen that to the membrane lipids somewhere the carbohydrate chain, which was represented in green, was shown there, right? So it is composed of phospholipids, cholesterol, and glycolipids. Now, phospholipids are the most abundant lipids in the membrane, and they spontaneously aggregate in such a way that they bury the hydrocarbon tail into the cell interior, making the exteriors of the membrane highly hydrophobic, while the interior is making the extremes of the membrane hydrophilic because of the phosphate head groups, and the interior highly hydrophobic because of the hydrocarbon tails that are embedded there. So if we take a look at the phospholipids, right, you can see the structure here of a phospholipid, and you can see the charged phosphate head here, and these are the hydrocarbon tails. So all cells have plasma membranes, and many of their organelles also have membranes. We have talked of the endomembrane system, right? In the earlier classes, we have been talking about the cell organelles, where you have seen that they are also membrane bound. And all membranes are made up of a bilayer of phospholipids, or sometimes a monolayer. Now, the hydrophilic and hydrophobic parts you can see here. The phosphate head is hydrophilic in nature, whereas these hydrocarbon tails are actually water repelling or hydrophobic. Then the arrangement of phospholipids in membrane. This is the phosphate head part, and this is the hydrocarbon tail, right? So the cell membrane has two layers of phospholipids as shown here, and the hydrophilic heads are facing an aqueous environment. One is the cytoplasm and other is the extracellular environment, whereas the hydrophobic tails are facing one another, as you can see here, right? So these are the phosphate heads, and this is the hydrocarbon hydrophobic interior of the membrane. Now, once you take a look at the phospholipid bilayer, right, just feel like these lipids are fixed in places. It is not like that. Right? The phospholipids have the ability to move laterally. Okay, these movements are not very frequent; they are rare, but they can make a turn of 180 degree and can exchange places. Okay, so these are called the lateral movements, where adjacent phospholipids can change places. Now, other than this, the lipids can also exchange places in between the bilayers also, right? This is called the flip-flop movement. So, in short, we can say that the membrane phospholipids actually show two types of movements. One is lateral movement, right, which is these phospholipids exchanging places with adjacent lipids, and this movement occurs within one layer of the you can say within the same layer of phospholipids. And the other is the flip flop movement, where lipids can exchange places in between membranes. Now, this lateral movement is very important because this only imparts. We have talked that these phospholipids are self-sealing. If these movements will not be there, there will be no flexibility in the membrane. Now, this flip-flop movement is important because you see, where did I tell you that the lipid synthesis takes place in a cell? Once we have talked of different cell organelles, I've told you about the organelle which participates in lipid synthesis. Okay, right, Diksha, you have talked of the smooth endoplasmic reticulum or the endoplasmic reticulum. So this means it will be present in the cytosol. So all the phospholipids will also be synthesized there, right? So if Now, phospholipids are a component of the external membrane also, and I've told you that during endocytosis, this membrane has tendency to form vesicles and cell material into the cell interior. So this means these lipids have to be constantly replaced. So, because of these flip-flop movements, all the lipids that are synthesized in the cytosol, they first enter into this cytosolic side of the lipid bilayer or the internal lipid bilayer, and they are finally, through flip-flop movements, transported to the cell uh, exterior side of the lipid bilayer. Otherwise, also these lipids can exchange places when required. Right. So, did you get this? What are these two movements, and what is their significance in the plasma membrane? Okay, fine. Let's move ahead. Then we will talk about the nature of phospholipids, right? That is, we have to talk about the saturated versus the unsaturated phospholipids. I hope you know the meaning of saturation and unsaturation in terms of bonds. Yes, please tell me. Okay, I think Shraddha is posting a response. What is the meaning of this term saturated and unsaturated? Okay, Palak says saturated means single bonds and unsaturated means presence of double or triple bonds. Right. Okay. So it is seen that depending upon the degree of saturation and unsaturation in the hydrocarbons that form the phospholipids, it actually directs or decides whether the membrane is going to be fluid or is it going to be viscous. It has been seen that membranes which are rich in saturated hydrocarbon tails containing phospholipids, these are more viscous, whereas those which contain unsaturated hydrocarbon tails, due to unsaturation, it is seen that there is a kink wherever there will be an unsaturated hydrocarbon in this tail. There is going to be a kink, and this kink actually creates a gap in between the adjacent phospholipids. So that is why it incorporates more gaps in these lipids. So it is seen that membranes are more fluid when they contain more unsaturated fatty acids within their phospholipids, and more unsaturated fatty acids result in increased distance between the lipids, making the layer more fluid. So this is important to understand. Did you get this? Everyone, Chavi, Diksha, Inakali, Niharika, Palak, Ritu, Shraddha. Okay, fine. Let's move ahead. Now let's come to the proteins, right? Now these lipids actually form the structure, right? Jesse, if for example, if you take into consideration the outermost boundary of a house, you'll always see it might have one gate or two gates that allow the movement of a person or some car go through it. So in the same way, these lipids, they actually form the boundary, and these proteins are actually the functional components of the membrane that help the membrane perform its different functions and help it communicate with the extracellular environment. So It is a mosaic of proteins, means it contains a variety of proteins that actually impart different properties to the membrane. So membranes are very complex and dynamic, containing many different types of proteins, and these proteins are stuck in the membrane like a mosaic. Proteins actually decide most of the functions of the membrane, and it is the different proteins that are responsible for the uniqueness of different membranes. I mean to say, the type of proteins, its functions, you see each and every cell has a different function. If we compare a prokaryotic cell with a eukaryotic cell, and within eukaryotes also, if you take different plants, different animals, you'll see they contain different type of cells in a single living organism also that can perform different functions, right? So their membranes are different, they contain different type of proteins in different compositions that give uniqueness to each and every type of membrane that is present in different living system. So the collection of proteins in the membrane varies from membrane to membrane, that is from one type of cell to another. Now, if we talk of the membrane proteins, then we can categorize the membrane proteins into two basic types. First one are the peripheral or the extrinsic proteins. Okay, I'll just go back to the diagram, right, of the membrane. Okay, you can see here there are certain proteins that are located to either side of the lipid bilayer, right, and there are some proteins that are embedded actually. Okay, so these embedded proteins are called integral proteins or transmembrane proteins, as they can cross both the lipid bilayers and connect the cytoplasm to the cell exterior. Whereas these proteins, which are located only to one side of the membrane, as you can see here, that are not embedded, okay, here, these are called the peripheral proteins. So let's talk of proteins again. Now, if we talk of membrane proteins, we can categorize them into two types. First one is the peripheral or the extrinsic proteins, whereas the other one is the integral, also called the intrinsic proteins. So peripheral proteins are not embedded in the lipid bilayer, and they are loosely bounded to the surface, right? And if we talk of the intrinsic proteins, they penetrate and often completely span the lipid bilayer, right? So they are also called as transmembrane proteins. Where they contact the core, they have hydrophobic regions with non-polar amino acids. Now you know proteins are formed of amino acids. So it has been seen that since the membrane is amphipathic in nature, it has a hydrophilic end also, and it has a hydrophobic part also. So it has been seen that when these transmembrane proteins or intrinsic proteins, they cross both the lipid bilayers. So the part that actually comes in contact with the hydrophobic interior is rich in hydrophobic amino acids, whereas the ends of that protein that connect to either the cytoplasm or the extracellular side, they have hydrophilic regions. This is important because if this is not the case, then the protein will not be stable in the lipid bilayer, and that is going to affect the integrity of the membrane. So we have peripheral proteins and integral proteins. Now, if you wish to separate these extrinsic proteins from the membrane, they are loosely bound to the membrane. You can separate them easily without destroying the complete structure of the membrane. But if you have to separate an integral or intrinsic protein from a membrane, then definitely you have to break off the membrane integrity, because they are very tightly bound to the membrane. All the receptors, all the channels, all the transporters that you'll see in membrane, once we'll talk of the different functions of the membrane, you'll see these are actually these intrinsic or the transmembrane proteins. So is this clear to all of you? What are peripheral proteins and what is their significance, and what are intrinsic or integral proteins and what is their significance? So you can see how a membrane has different variety, and now a membrane can have different type of peripheral proteins and different type of integral proteins. Okay, let's move ahead. Now, we will be talking about the functions of the plasma membrane in detail in the subsequent classes, but I just want you to take a look at this picture, right, where you can see why different type of proteins are required and how the presence of different type of proteins helps membrane perform different functions. So membrane protein can help the plasma membrane in transport. You can see it can help either in active transport that works with the utilization of ATP or energy, or it can allow passive transport of molecules also across the membrane. Then these membrane proteins can act as enzymes also, right? These proteins act as enzymes, and they can lead to different cytosolic or initiate different cytosolic reactions by their domains that connect them to the cytosol. Then these proteins help in signal transduction. These membrane proteins actually act as receptors that can receive ligand, or you can say signaling molecules from the extracellular space that can come and bind here to the membrane of a cell and transmit a message to the cell interior that can bring about any, you can say, biochemical or morphological change in the cell. So they help in signal transduction also. Then you can see they help in intercellular joining. I've told you about the cell adhesion proteins, right? Where you will see in a tissue, different cells are joined together by these adhesive proteins. So two cells can be joined together through these membrane proteins. Then cell to cell recognition. This I've already told you that these glycoproteins, you can see if you take a look at this membrane, it has a carbohydrate attached to its protein that forms or makes it a glycoprotein. And this is important in, say, for example, antibody responses. Suppose a particular, immune system cell has to go and identify, say, a cell that has been attacked by a pathogen. So in that case, it is seen that that particular tissue changes the behavior of its membrane glycoproteins. So this can help one cell to recognize another cell by the pattern of this glycosylation. So thus, the membrane proteins also help in cell to cell recognition. Then of course, once I had shown you the complete picture of the membrane, I had told you about the extracellular matrix. So these proteins actually connect that particular cell to its extracellular matrix, which is very important. Again, we'll talk of the extracellular matrix in detail, but its connection to the extracellular matrix is important in signaling, in protection, and in various other functions. So attachment to the cytoskeleton and the extracellular matrix of a cell is also through its membrane proteins. So now you can see why a membrane is said to be a mosaic of these proteins. A particular cell can have all these proteins, different type of proteins in its membrane in each and every cell. So it has a diversity
Okay, so
of
this is a video
proteins
about the
that can perform diverse functions also. So is it clear to all of you? I hope you agree that why this mosaic fashion of protein is important in the structure of the membrane. Because if it is not going to be so, then the membrane will never be able to participate in all these functions. And you know, each and every cell has its barrier, its plasma membrane. In order to keep it connected even to its adjacent cell in a multicellular system in a tissue, it has to have its membrane very active and very functional. Okay, let's move ahead. Now we have to talk of the last component, that is the carbohydrates that are present in the membrane. So the carbohydrates actually form the glycocalyx, right? I've told you that the carbohydrates are always present to the extracellular side of the membrane. And if through an electron microscope, if we take a picture or if we take a look at that surface of the membrane, it seems that a layer of carbohydrates is present just above those lipids or those proteins. I mean, it appears to be like shading the membrane. That part is called the glycocalyx. So the glycocalyx of the membrane is actually formed by its carbohydrates, which ultimately are either attached to the lipids or to the membrane proteins. Now, as I've told you earlier also, these are used for cell-to-cell recognition, that is the ability of a cell to distinguish one type of neighboring cell from another. This forms the basis of immune responses, and these membrane carbohydrates, whenever present, they are usually branched. They are not linear. You have seen in the structure also. Now, the pattern of glycosylation and this glycocalyx, its properties, it varies from species to species, individual to individual, and even from one cell type to another within the same individual also. That is, for example, you can see the blood groups. It is because of these membrane carbohydrates only. So different type of carbohydrates are present, and they actually impart different type of properties to the membrane. Okay, so I think this is all about the basic structure or basic components of the membrane that I had to tell you. This is just the introduction. In the in lectures, in our further lectures, I'll be talking to you about what are the different type of phospholipids that are present in the membrane, and then we'll also talk of what are the different type of functions that a membrane performs, and especially we have to talk about the transport across membrane, how these different type of proteins help a membrane connect its cell interior to the extracellular site. Thank you so much.